Hey everyone, welcome to Professor Long's Lectures in Anatomy and Physiology. I'm Professor Long. I'm going to be covering some biochemistry with you guys today for my Human A&P2 class. As you guys know, we're in this coronavirus shutdown, so we're converting from face-to-face to online, so this is how I'm delivering my instruction. This video is really directed at students taking my Human Anatomy and Physiology class and the metabolism lectures that we need to cover. We've already done the first metabolism lecture, and at the beginning of that lecture, I kind of gave you, I was hoping to get it all done in one, but I couldn't because I really did need to back up and cover some information for y'all. This is metabolism lecture number 2 for my Human A&P2 course. If you're following along in my note set, the published note set, you can follow along on page 86. Okay, if you're not in my class, I hope this helps you understand some information. Other instructors, both at our college and at other colleges and universities across the U.S., might teach this slightly differently. My approach is that a lot of my students don't have a lot of strong chemistry background, and so I'm going to approach this from the simplest explanations I can possibly give without getting too complex into the chemistry, okay? Because you can really lose some people in all of this. So my lecture might be a little simpler than some people's, but that's okay. My goal is for you to understand why we're undergoing what we're going, or why we're learning what we're learning. Where does it fit in our human physiology? So what we learned in the last lecture is that our cells, and I'm going to erase this, but I'm going to run it real quick, can take glucose in the presence of oxygen, run them through mitochondria, and we get 36 ATP. That's a lot of energy. That's the big deal here. Up to 36, excuse me. And then we're going to get some water, and we're going to get some carbon dioxide, okay? Oddly enough, plants take water and carbon dioxide and convert it into glucose, C6H12O6. They need the energy of sunlight, photosynthesis, to do that. Anyway, so now, this is the setup. How do we get from here, a glucose molecule, to 36 ATP? And one of the things we can do is we can convert glucose into 2 ATP without all this other stuff. That's not enough energy for our bodies. That's great for bacteria. Bacteria do this, and some other simple organisms, because they don't have all that actin and myosin, every muscle contraction burning billions of molecules of ATP. They don't have brains and nervous systems conducting action potentials with ion pumps and things. They're not burning much energy. They go wherever the wind or water blows them. If they land on a food source, they survive and reproduce, and if they don't, sometimes they can go into a spore state and quit burning energy. We need tons of energy, so we have to have this to survive. Okay. Now, let me erase all of that. That's the kind of the setup, what we're aiming for. Now, on my board here, I'm going to draw one giant cell. Okay. If I were you, if you're going to try to draw this, turn a page sideways and draw the cell and just leave a little bit of border around the edges here, just like I'm going to do on the marker board. Okay. So I'm going to have a giant cell here. It's going to take up most of my marker board, but not all of it. Okay. This is our cell membrane. This is the lipid bilayer. And in my cell, I'm going to draw one giant organelle. And that one giant organelle is going to be a mitochondrion. And it turns out that mitochondria have two membranes, two lipid bilayers. There's an outer mitochondrial membrane and an inner mitochondrial membrane. Before I get too far, I think it's kind of interesting. Ancient cells had a cell membrane, and there's these ancient archaebacteria, so to speak, an ancient organism that has the exact same lipids and proteins in this membrane that are found in this membrane. And this membrane has the same lipids and proteins as this membrane. And it turns out that at some point, our cells, eukaryotic cells, engulfed this organism into a vesicle, but the lysosomes didn't digest it. There's a symbiotic relationship here. Little biology. Symbiosis. Sym means with, and bios means to live. They are mutually beneficial. When you have a symbiotic relationship, our cells are giving them things that they benefit from, and they are in return giving our cells things that they benefit from, and so they live together rather than one destroying the other. If this cell destroyed, or if the organelle destroyed the cell, that would be called a parasitic relationship. In parasites, the host is hurt by the parasite. The parasite gains, but it actually takes away from the host and can kill it. Just like human relationships. You ask yourself, are you in a symbiotic relationship or are you in a parasitic relationship? If it's parasitic, man, it's going to drain the life out of you. Anyway, so now, that's why mitochondria have two membranes. The outer membrane is identical to the cell membrane of our cells. The inner membrane happens to be same membrane as this ancient organism. Now, that's not important for you to take notes on, so if you wrote on your picture, I'm sorry, but anyway, that's mitochondria. Now, the substance inside the mitochondria, so we have the outer mitochondrial membrane, there's an intramembranous space, and the inner mitochondrial membrane, and then this fluid in here is very often referred to as the matrix, okay? M-A-T-R-I-X, matrix. Now, I'm going to erase the matrix because I need to write inside of here. And I'm actually going to rearrange or draw the inner mitochondrial membrane a little bit smaller because I need some space inside of here. Okay? So if you're drawing yours, you might want to redraw it. And actually, you should practice drawing this till you can draw it from memory. But the inner mitochondrial membrane is all folded up. Okay? Each fold is called a crista. And cristae, with an E on it, would be plural. So these are called mitochondrial cristae, the folds in the inner mitochondrial membrane. That's an important term because it is on those cristae that we have a series of enzymes that are all kind of linked together. There's a series of molecules here, and the more folds, the more of these enzymes. And some of these molecules, there's a stuff called coenzyme Q, and there's some other enzymes in here, but some of these things are coenzymes as well. Some of these molecules in here are referred to as cytochromes. So I'm going to give you the name of them. They're called cytochromes. Those are the proteins, many of the proteins that are embedded on the cristae of the inner mitochondrial membrane. Those cytochromes end up being sort of enzymatic, so to speak. They're going to play an important role in our energy metabolism. So this is the extracellular fluid outside the cell. This is the cell cytoplasm. And then this is the matrix of the mitochondria. And the inner mitochondrial membrane has all these cristae on it, okay, and cytochromes. Now that we have the terminology down, here's what goes on. When all of us eat sugar, and we digest it, we get these glucose molecules, C6H12O6, but I'm not going to write out the chemical formula. It turns out that in our cell membranes, we have some little channels called glucose channels that will let glucose into the cell. If the glucose channel is closed, then the glucose remains outside the cell in the extracellular fluid and in our bloodstream. What opens the glucose channel is that when our liver, and really there's more involved like with the stomach, but and gastric inhibitory peptide from the pancreas. But essentially, when the pancreas, I said liver, when the pancreas detects glucose, then the beta cells of the pancreatic islets, the islets of Langerhans, can release molecules called insulin. I'm going to put a little I with a circle around it. Well, it turns out that the glucose channels have insulin receptors on them, and it takes 2 molecules of insulin to open one of these glucose channels, and now glucose can enter the cell. This is why insulin is important for reducing blood sugar levels and allowing insulin uptake, glucose uptake, glucose being taken into our cells. If you lack insulin, then you have type 1 diabetes, and you have to inject it. If you lack the receptors, you have type 2 diabetes. It's non-insulin dependent. Nonetheless, that's how we get glucose into our cells, into our cytoplasm. Once it's in our cytoplasm, glucose is going to undergo a whole series of biochemical reactions. And at the very end, I'm going to get 2 molecules of this stuff called pyruvate. Okay? And all of this, I'm going to use an NZ for enzyme reactions. These are all enzyme reactions that convert glucose into 2 molecules of pyruvate. Pyruvate is carbon, hydrogen, and oxygen, but it's only 3 carbons. Now, in order to start this whole process, it's going to require my cell to take in or burn 2 molecules of ATP. I need to burn a little energy in order to make more. It's kind of like investing money. You give someone $2, if you get $4 back, you've gained money. So at the end of all of this process, what I'm going to get out of here is a couple of major compounds. Okay, one of the things I get out is I'm going to get 2 molecules of ATP. Actually, I'm really going to get 4 molecules of ATP, but there's a net gain of 2. I gross 4. But because it cost me 2 here, I only gained $2 or 2 ATP. Okay, but that's a big deal. So this process and the breakdown of glucose into pyruvate is called glycolysis. And there's going to be 3 steps to the energy metabolism in glycolysis. What you need to know, and all of this is written on the bottom of page 86 of my note set. In glycolysis, it occurs in the cytoplasm of our cells. That's where the enzymes are. We convert glucose into 2 molecules of pyruvate, and we get a couple of important things out of it. We get 2 ATP, so we've converted glucose into energy, and we're going to get these other substances out of it, and I'm going to try to write them in a different color. I'm kind of running out of colors here, but let's see if pink shows up on the screen. What I'm going to get, can you see that? Yeah, you can. What I'm going to get are these molecules called NADH, and I actually get 2 of them. Now, I'm going to do a little side note here. There's a molecule called NAD+, nicotinamide adenine dinucleotide. You don't need to know that, but NAD has a plus on it because it's missing an electron. Now, as I told you on the last lecture, all of these atoms that are making up these molecules are made up of smaller bundles of energy or condensations of energy, protons, neutrons, and electrons. And when we're forming molecules and forming atoms and breaking them apart, it takes energy and releases energy when we're putting them together and breaking them apart. And I don't know if this has ever happened to you. I'm sure it has. You ever take something apart, like a dresser, or you're building a barbecue pit or something, and then when you're done, you notice there's like 2 screws, a spring, and a washer left over. And you know they were in there when you took it apart, but when you put it back together, you're like, so you just scoop them in a drawer and hope it doesn't blow up on you or something. Well, when we rip atoms and molecules apart, sometimes we have some extra electrons and protons hanging around. And when we're ripping this apart and putting it back together and combining it with other substances to make pyruvate, we have some extra electrons left over. And if you remember, a hydrogen atom, a hydrogen ion, is nothing more than a proton. And if I combine it back with its electron, I get hydrogen out of it. If I break hydrogen apart, I get a proton and electron. If I put them back together, I get a hydrogen atom. Well, guess what? I got an extra proton there, and I have some extra electrons when I'm ripping this apart that are left over. So if I stick those electrons onto NAD+, I get a molecule called NADH. So essentially, this substance called NAD+, comes over here, gets some electrons, and it's going to carry those electrons somewhere else in the form of a hydrogen ion. So we call this a hydrogen ion carrier or an electron carrier. So some books will call it an electron carrier, and some people say it's a hydrogen carrier. And that's the importance of NADH. We're going to get 2 of those suckers, but I'm not going to get into the math of this. So this is a big deal, the production of the NADH molecules. So let's review real quick before we go on to the next step. In glycolysis, you need to know it occurs in the cytoplasm of the cell. You need to know that when we take glucose in, we have to burn some ATP. We run the glucose through some enzyme reactions and end up with 2 molecules of pyruvate. Out of that, I'm going to get 2 important things. I'm going to get 2 molecules of ATP. I'm going to get a couple of NADHs, or electron carriers, hydrogen carriers. This is going to be important in just a little bit. Got it? Now, the importance of pyruvate. And if you don't know this yet, again, I'm going to make a little side note here, and I'm going to erase it. So one of the things that happens is when we have an acid, let's say I have a compound that has a carboxylic acid. Carboxylic acid to a chemist means a carbon with a double bonded oxygen, and a hydrogen here. As you know, this acid can fall apart, and what I'm going to get is the negative ion plus the release of the proton, because acids are hydrogen ions or proton donors. This is the 8. I've gone from pyruvic acid to pyruvate. Anytime you see the 8 at the end of a word in biochemistry, it means the negative ion of an acid. It's given off the hydrogen. So sometimes we'll say you get 2 pyruvic acids or you get 2 pyruvates, because it will dissociate into pyruvate. Now, what's important about that pyruvate is that's sort of like the leftovers that normally in bacteria and other things can go into other chemical reactions, and we don't do much with it. Our mitochondria have the ability to take the leftovers, sort of the trash, and it's sort of a reduce, reuse, recycle. We're going to reuse this to get more energy out of. Because other organisms like bacteria and prokaryotes don't have mitochondria, that's the sum total of energy that they get from one molecule of ATP. We're going to get more out of it because of our mitochondria. So here's what happens, okay? I'm going to take this pyruvate, and I'm just going to put a P with a little circle around it for my pyruvates, okay? These pyruvates are going to go into the matrix of the mitochondria. And as I take the pyruvate in there, it's going to get converted into another molecule called acetyl coenzyme A, or acetyl-CoA. And in that process, it's going to give us some important stuff. But essentially, this kicks off a whole series of chemical reactions. And as these chemical reactions occur, there's going to be all these changes, and then we end up back with acetyl-CoA. And all I'm going to do is rip the carbons and hydrogens and oxygens apart and combine them with other things in a series of chemical reactions and then put it back together. But in that process, what I'm going to get out of it is a couple of things, okay? And this, by the way, is going to be the second step in our energy production. First one is called glycolysis. The second one is called the Krebs cycle, after a guy named Sir Hans Krebs. He was knighted for all of his biochemistry that he figured out. It's also called the citric acid cycle, because we convert acetyl-CoA into citrate, isocitrate, citrate, citric acid, isocitric acid, citrate, isocitrate, malate, fumarate, alpha-ketoglutarate, succinate, succinyl-CoA. I forget the order. If you give me a second, I'll come up with it, but it doesn't matter. All of these different intermediaries, we're just manipulating acetyl-CoA and converting it into all these other things, and then we end up back at acetyl-CoA. We can bypass that if we just inject citric acid. We can skip all of this. That's why citric acid orange juice is a very quick way to produce energy. If you're ever really low on your blood sugar, drink some orange juice or something high in citric acid like lemonade or lime juice and stuff, and you'll get your citrus fruits. So it's called the citric acid cycle because it uses citric acid in here. And it's also called the TCA cycle. TCA stands for tricarboxylic acid. Citric acid has 3 carboxyl groups on them, carboxylic acids. So it goes by all 3 names: the Krebs cycle, the citric acid cycle, or the TCA or tricarboxylic acid cycle. You'll see all 3 names used. So what you need to know is, what is the importance? What happens here? In the TCA cycle, we convert pyruvate into acetyl-CoA, run it through a bunch of enzyme reactions, and we're going to get some important stuff out of it. One of the things that it kicks out is every time the first pyruvate goes around, I get a molecule of ATP. And when the second pyruvate goes around, it has to go around twice, one for each pyruvate. Each pyruvate will give me an ATP molecule. So now I've doubled my energy production. I've got 2 ATP from glycolysis and 2 ATP from Krebs cycle. Still not enough for us. And then finally, the other thing that gets kicked out here is some molecules called NADH and FADH2. It's called FADH plus. When I stick an electron on it, it turns into FADH2. And again, these are electron carriers, or we could say hydrogen carriers, okay? Same as this one. These molecules are super important. The last thing we get out is one of our waste molecules. We get stuff out of here that we don't really want, and I can't find one of my markers. Ah, there it is. One of the things that comes out of the Krebs cycle is the carbon dioxide. Now, we could count how many carbon dioxides and how many NADHs and FADH2s come out of each step, but I'm not going to get into the numbers. You know, you can read that in the textbook. I want you to understand the purpose of this. Glycolysis converts glucose into 2 molecules of pyruvate, 2 ATPs, and some electron carriers or hydrogen carriers in the form of NADH. The pyruvate gets recycled by the matrix inside the matrix of the mitochondrion, converted into acetyl-CoA and then citric acid, and it goes around and around and around, hence the term the citric acid cycle or the TCA cycle. Or Sir Krebs figured it out, Hans Krebs, so it's called the Krebs cycle. The importance of the Krebs cycle is this. It doubles my energy production, it gives me more proton and electron carriers, and then I get some carbon dioxide. This is where the carbon dioxide comes from energy production, and we have to exhale that carbon dioxide. Finally, if I go look at all of these little proteins that are in the inner mitochondrial membrane called the cytochromes, those cytochromes are going to play an important role. And I'm sorry, give me 1 second. I'm not going to kill a 20-minute video because my dog wants in. I apologize for the interruption, but I'm at home doing these videos for my students, and my dog wants in and out and in and out and in and out and in and out. Anyway, the last of the 3 steps, the third step, is going to be called electron transport. Let me fit this in here. The third step is going to be called the electron transport system, or ETS. Some books call it the electron transport chain because there's a chain of these proteins where we're going to transport electrons around. So some books call it the ETC, the electron transport chain. The electron transport system, or the electron transport chain, is also referred to, and this is an important term, so it's also called oxidative phosphorylation. Okay? I couldn't fit it on the edge of my board here, but oxidative phosphorylation. To phosphorylate means to stick a phosphate group onto something. And oxidative means it's going to be an oxidizing reaction. Okay? It's going to use oxygen in it too. So, now this is what happens. This is the big deal. This is the big finale here. All of these electron carriers, or hydrogen carriers, are going to carry those electrons and hydrogens to these cytochromes in the mitochondrial cristae. And what those things are going to do is they're going to accept these electrons, these little bundles of energy. And it's almost like if I walked over to a student. Let's say I had seven students in a row here, and at the end of the row there's sort of a bucket of water. And I handed one a steaming hot baked potato. That's going to burn, so they're going to transfer that to the next one, to the next one, to the next one, and then the last one's going to dump it in some water, and that can handle it. That can take the energy, the heat that's in the potato. Well, y'all would be the cytochromes. The potato is going to be the electron that's going to get transported from one to the other, and then we're going to combine that electron with some oxygen molecules and form water. And essentially, that's what happens. So I know this is a big mess, and they don't go outside the cell here, but essentially, all of these guys, all the NADHs, I'm going to transfer these NADHs over here. They would be transported into the mitochondria, and then what happens is we're going to rip electrons off and pass them back and forth, and eventually oxygen that we inhale is going to come by here, get combined with some of these protons, and form water. And in the process, one of those little cytochromes that is going to convert oxygen and these electrons, or the protons and the hydrogen atoms, into water, and it's some complex chemistry that we don't have the time for. It really would kind of melt our brain. It still melts mine to this day. But nonetheless, what that's going to do is that's going to kick off between 30 to 32 ATP molecules. I'm going to go with 32 because that's the way that I learned it. So, and really all it does is it takes a molecule of ADP and some phosphates, and when this protein takes the oxygen and the electrons and combines them to form water, it also takes ADP and phosphate and puts them together to make ATP and spits it out. It's the opposite of when we burn energy to do something. It's actually forming energy, and this is where most of the ATP in our energy production occurs, at the cytochromes in the mitochondrial cristae, in the electron transport system, the electron transport chain, or during what we call oxidative phosphorylation. I'm using this oxygen to phosphorylate a molecule and add phosphate to it. So all of this is on page 86 and page 87 of my note set. And really what you need to know is this information down here at the bottom of the pages. What occurs in glycolysis and where does it happen? In glycolysis, it occurs in the cytosol of the cell. We convert glucose into 2 molecules of pyruvate. We get 2 things out of that. 2 ATP, a net gain of 2 ATP, and some hydrogen carriers for electron transport system. The pyruvates each go into the citric acid cycle. They get converted into acetyl-CoA and then citric acid. They go around and around in a cycle here. And when one pyruvate goes around, I get 1 ATP. When the second pyruvate goes around, I get another ATP. So now I have 4 ATP total. I get some more hydrogen carriers, and I get some carbon dioxide. That's where the carbon dioxide is produced, comes out into the cytosol, dissolves into our plasma, and then we exhale it when that passes through our lungs. Now, the big kicker here is in oxidative phosphorylation. I take all these hydrogens from the hydrogen carriers or electron carriers. They donate some electrons and some hydrogens through these cytochromes. We take in oxygen to form water, and we get out 32 ATP from that process. Okay, that's where most of the energy in our metabolism, our energy metabolism in our cell comes from, is oxidative phosphorylation. It occurs on the cytochromes on the mitochondrial cristae. And all of that is written in my note set. And then there's a bunch of review questions here. And I think there's 15 or 16 review questions. And there's usually somewhere between 15 and maybe 20, 25 questions I'm going to come up with between the last 2 lectures. You need to know the definitions: metabolism, catabolism, anabolism, substrate, catalyst, active site. You need to know that sugars can be converted directly into energy. Lipids have to be manipulated but can be converted into energy. Proteins have to be deaminated and converted into energy. And then nucleic acids. We did that in the last lecture. And then what's really happening is once we get everything converted into glucose, we go through these 3 steps and produce 36 ATP. 32 plus these 2 is 34, plus those 2 is 36. That's where the energy comes from. If I cut off the oxygen supply, these enzymes shut down, which backs this up, and it shuts down, and I'm only left with the ability to produce 2 ATP. My body would burn that before it can make enough energy, and I would die. So we have to have oxygen. If you cut someone's oxygen supply off, you shut down all the biochemical reactions and kill them. Can't have that. Okay? All right, so now I hope you learned something. I hope you had as much fun as I did, and I hope this was helpful. I also hope to see you on the flip side in the lectures on the urinary system. So thanks for watching. See you next time.